Okay, so you may have noticed that I emailed you a decision tree. So just a piggyback. Turn the volume down there. Okay, hopefully I'm not getting too much feedback there. Piggyback on just mentioning that drop date is coming closer. I emailed you that decision tree just to, if you're thinking about it, that may answer some questions. I also want to reiterate that you can stay in lab, but you have to get your lab professor's permission to do that. So if you do drop 212, they will automatically drop you from 213 unless you get your lab professor's permission. You have to, a course override has to be generated. So just keep that in mind. Again, not trying to sway you either way. See me if you want a little help making that decision. Let's continue with our antibiotics. So I threw a lot of different drugs at you last time. I've got a few more that I want to talk about, a few different mechanisms, and then we'll get into talking about resistance a little bit. So let's see, we finished up last time with our protein synthesis inhibitors. That was a pretty large class of drugs, including the aminoglycosides, the tetracyclines, the macrolides. We just have a few more different mechanisms. Rifampin, or sometimes it's called rifampicin, same thing, is a RNA inhibitor. It actually blocks that enzyme of transcription, RNA polymerase. That enzyme in bacteria is different enough from our RNA polymerases that it's selectively toxic. It's semi-synthetic, and I don't know what part of that molecule is synthesized and what part is a natural product. It's a pretty complicated molecule there. But it gets absorbed pretty easily into the tissues in our body that need access to it. So it can be given orally or by IV or injected, either one. This drug has kind of the very interesting side effect of turning bodily fluids kind of a reddish orange color. That's just because the drug itself is that color. So again, it's sort of a uncomfortable looking side effect, but it doesn't really have any, problems associated with it. There's a catheter bag of somebody that's on rifampin in a hospital. So again, it's not really a problem. It's just kind of weird looking. There are some other side effects associated with this drug, like nausea and vomiting, kind of loss of appetite. We could use this drug for a lot of different things. I think it's fairly broad spectrum, but we primarily reserve it to use for TB and leprosy. A couple of infections that we don't have a lot of options for. So along with, remember the other drug that we usually use for TB, isoniazid. That was the mycolic acid synthesis blocker. Okay. All right, and then a couple of drugs that act to inhibit DNA replication are the quinolones and the fluoroquinolones. So the quinolones are kind of the early generation drugs. And then we kind of monkeyed with that structure a little bit, added a fluorine here, and that gave us the fluoroquinolones. So the early generations, the quinolones, the very first one was nalidixic acid. And our lab and lecture don't line up exactly here. Your next lab, you're going to be using a medium to grow bacteria on that has nalidixic acid in it. And that makes the medium selective because nalidixic acid only works against gram negatives. So inhibits gram negatives. Okay, so it's part of a selective media. When we're trying to grow only gram positives, it gets rid of the gram negatives. So again, that doesn't look familiar yet, but if you're in lab, that will look familiar later on. Some of the later generation quinolones were a little bit more broad spectrum, but that first one, again, only worked on gram negatives. Both the quinolones and the fluoroquinolones target DNA gyrase. So remember, back when we talked about replication, if I can draw, this is supposed to be a replication fork right here. And upstream, that DNA gyrase works upstream here as those two strands of DNA are getting pulled apart. Upstream gets kind of tangled up, supercoiled, so that DNA gyrase cuts both strands, unwinds it, and seals them up again. Okay, and that enzyme actually is slightly different between gram positives and gram negatives. And especially in prokaryotes, it's a little bit different than for eukaryotes. So again, selective toxicity. The fluoroquinolones, this fluorine atom here actually gave the quinolones a little bit more broader spectrum. It made it absorb better, and just in general was just a little bit better drug. Very stable in acid, so again, that can be given orally. We usually, so what kind of drug? An example of a fluoroquinolone is ciprofloxacin. That's probably the one that you've heard of, Cipro. We often call that, but all of these drugs end in that oxacin suffix. I always have to think about what's the difference between a prefix and a suffix. Okay, so they end in that oxacin, and there's a whole bunch of them. We could use this pretty much on any infection because it is so broad spectrum, but we usually reserve it for infections that are a little bit more serious when other drugs haven't worked first. I don't know, anthrax, respiratory infections. Again, usually when something else hasn't worked first, then we might prescribe a fluoroquinolone, because there are some kind of rare but nastier side effects associated with these drugs. A kind of odd one is tendon disruption in older adults, especially the Achilles tendon. So it seems like a very odd side effect. We're not really sure whether the drug kind of impedes blood flow to joints or whether it impedes collagen production. Not really sure. Again, fairly rare, but that's something that we see when it's given to very young children. It's been, again, rarely, but caused seizures in infants, possibility of nerve damage, some neurological stuff. So that's not good. Aortic rupture, obviously, that's bad. So again, we reserve this for when other things haven't worked typically. Oh, and sometimes when I find a mnemonic, I like to pass it on to you. So whatever, if this helps you, this mnemonic, the queen, that's supposed to stand for quinolone, stops the gyrating dancers. So gyrating DNA gyrase, that's the target. Okay, so whatever, maybe that helps, maybe it's confusing, but sometimes you get a picture in your head and it can help you remember stuff. Another class of drugs, which there are only 2 examples right now that are in clinical use right now, and these are cell membrane inhibitors. They're called the polymyxins. Colistin is another name for polymyxin E. Okay, I think colistin is the trade name or the brand name, and then the drug is polymyxin E. That's another one that you're going to use in lab next week, so put that up there as an example. The structure here, you can kind of see a ring and then this tail. It kind of acts in a way similar to our soaps and detergents, how they kind of insert into membranes. And of course, anytime the plasma membrane of a bacterial cell is disrupted, that's going to be really bad for the cell. Works especially well against gram negatives. So they've got that outer membrane. The polymyxins can easily kind of insert through that very thin peptidoglycan layer and into the plasma membrane as well. When we administer polymyxins, usually we only administer this topically because big toxicity issues with the kidney. Okay, so for that reason, we only use this for skin infections, with the caveat that if the patient's life in danger and nothing else is working, then we might, again, administer this by IV if other drugs haven't worked. And I was reading about this yesterday that even sometimes they administer this in an aerosolized form for cystic fibrosis patients that have some kind of lung infection. So typically topically, but there are some other possibilities there. So if it's a topical drug, we're typically going to use this for skin infections. This is the third antibiotic in triple antibiotic cream. So along with, remember, we had a cell wall inhibitor, bacitracin. Our protein synthesis inhibitor was neomycin. And then we have, it's called polymyxin B. Forget what. Yeah, polymyxin B. So there's polymyxin E and polymyxin B. Those are, again, the two polymyxins that are in clinical use right now. All right, lastly, there's a big class of drugs called sulfa drugs that are inhibits a metabolic pathway of bacteria. So again, it's a big class of drugs called sulfonamides. The way they work is to target folic acid biosynthesis. So in bacteria, this is, don't memorize this pathway, but basically one of the substrates here in this pathway is called PABA, or para-aminobenzoic acid. And the sulfonamides act by inhibiting the first enzyme in this pathway. There's 1, 2, 3 different enzymes where you end up with tetrahydrofolic acid, which is essentially folic acid. Okay, and folic acid is necessary for the cell to make DNA. Apparently, folic acid plays a big role in stabilizing the DNA. It has a role into how and when it gets methylated. Folic acid is also an important cofactor for some of the enzymes involved in DNA synthesis. So it makes a really big role there. Okay, so the sulfa drugs inhibit this enzyme, the first one in that pathway. It turns out that there's another drug called trimethoprim that inhibits this enzyme, the third one in the pathway. So we usually put a sulfa drug together with trimethoprim, and then they work synergistically. They, one drug helps the other one work better. It's kind of a 1-2 punch. Like if the sulfa drug doesn't stop the process, then hopefully the trimethoprim will. Okay, so when we put those together, it's a drug known as SXT. So that's sulfamethoxazole, and then the T is for trimethoprim. Okay, so all the sulfa drugs have this sulfonamide functional group in there. That's why we call them sulfa drugs. So SXT, if you're in lab, you use that in the Kirby-Bauer tests when we did that last week. It has the brand name Bactrim. So that, I don't know, may or may not be familiar. We don't use sulfa drugs as much as we used to, but of course they came out before the rest, penicillin and other antibiotics. This figure just gives a little bit closer idea of what's going on here, or presents it in a little different way. So here's the substrate PABA, or one of them, that has to fit into the active sites of that first enzyme in the pathway. And again, don't worry about memorizing what that enzyme is. But here's the active site, right, where the substrate fits in. And then what you get eventually as a product, there's several other steps, but you get folic acid. Okay, so what if there is a sulfa drug nearby? And again, there's a lot of different ones, but you can see that all the sulfa drugs have the same, they mimic, they've got that benzene ring, just like PABA, and then the side chains are different. But if a sulfa drug, let's say, is close by, it's going to bind to the active site instead of PABA. Okay, so now it can't bind, and then you're not going to get any folic acid because not the correct substrate in there. So I think this was a question on the last exam, you know, gave you an example of a sulfa drug and how it binds to the active site. So this is an example of competitive inhibition. Okay, so no folic acid. So you can see here, you know, what's the so what? The bacteria is not going to be able to make DNA and RNA, or they're going to kind of be full of mutations, basically. Okay, so hopefully you're thinking about, well, how are sulfa drugs selectively toxic? And maybe your lab professor talked about this, but humans do not synthesize folic acid. Okay, we don't have that metabolic pathway. We get our folic acid from the food we eat. And in fact, we probably get a little extra folic acid from the bacteria in our guts that make a lot of folic acid. They usually make a little extra. So we get a little bit of folic acid from, you know, organisms like E. coli in our colon. I realized that I misspelled Gerhard Domach's name, but remember we talked about him. He was the German scientist that in the 1930s came up with the first sulfa drug. Again, that was a very narrow spectrum. It only worked against streptococcus, but again, that was a big deal at the time. They're a little bit more broad spectrum now. We don't use them as much because we have so many other antibiotics to choose from, and there's a little higher incidence of allergies. Sometimes a penicillin allergy will go kind of hand in hand with the sulfa drug. So we just have a lot of other options now, and they're just not used as much. But there are certain things that they might be more commonly prescribed for, like urinary tract infections, for example. But okay, still used a lot in vet medicine because they're really cheap. All right, so to kind of sum up a little bit, I filled in the rest of our concept map. I would encourage you to fill those things in at some point. It should be right in there in your notes. So this is a good way to just practice drawing this out. The most common thing that I'm going to ask you about is how these drugs work. Pay attention to any side effects that were notable. Anything, oh, we specifically use this for whatever. Okay, so that's what you're going to want to pay attention to. Okay. Right. So we can't not talk about bacterial resistance to antibiotics, because before the pandemic, this was really the #1 health emergency. Because if we run out of antibiotics that are effective against bacterial infections, that's really going to set us back in healthcare a long time. Anytime anybody has surgery, they're always taking antibiotics to make sure they don't get an infection before and after. So it's going to make surgeries a lot more, what's the word, dangerous. So we really important that the man that they Chain and Flory first used an antibiotic on, he just got a little cut, I think, on his chin from a rose bush. And he ended up dying from that because they couldn't make enough penicillin to treat him. So we don't want to go back to days like that where, we have to worry about every little cut that we get. So in general, how we're going to look at this much more specifically in some specific mechanisms, but how do bacteria become resistant to an antibiotic? The mutation rate is pretty fast because, right, bacteria are, they're reproducing so quickly. So they're going to have more mutations. With any population of microbial cells, you might have a few cells that by some mutation become drug resistant. And as long as, if they don't, usually if they're drug resistant, they might not be as fit in other ways. So oftentimes those cells might get out-competed unless they are in the presence of some drug. Then those drug resistant organisms are going to survive when the others won't, and they can grow up to become kind of the dominant strain or whatever. So when we talk about drug resistant organisms, it gets to be kind of a alphabet soup. We often give them abbreviations like MRSA, methicillin resistant, Staph aureus, or VRE, vancomycin resistant, enterococci. Pretty much almost any bacterial infection that you talk about, we worry about resistance. Gonorrhea, we've had to constantly change the antibiotics that we use to treat that. There are strains in Southeast Asia that are resistant to all antibiotics. Tuberculosis is another one, that huge problem. About 10% of new cases are multi-drug resistant, and about 1% of new cases are basically resistant to everything we have. So huge problem. It's expensive to make new antibiotics and to develop them, and pharmaceutical companies want to get their profits out of it. So it's a tough thing. So sometimes bacteria will get resistance just by random mutation. Oftentimes, though, they acquire the ability to make some kind of enzyme. And so that often is not just a random mutation. They're acquiring that ability through some kind of horizontal gene transfer. So remember our three kinds of horizontal gene transfer. There's transformation, can't see that very well, where bacteria might take up a piece of DNA from a dead cell. There's transduction, where a virus would accidentally maybe accidentally, or we can make viruses do this on purpose, actually, where they'll take some of one cell's host DNA and then infect another cell. And if it's a lysogenic virus, that DNA from one bacteria cell could actually incorporate into another bacteria cell. But we think most often that conjugation is the way that resistance plasmids are passed. So remember, if you have a F+ cell, that's a cell that can make that pilus, and that pilus will bring two cells together, a pore will open up, and the plasmid from the one cell can get transferred to the other, the fertility plasmid, as well as other maybe resistance plasmids. So, and that can happen between cells that are the same species or different species. So they can actually pass lots of information that way. How have we gotten to this point that we have so much problem with resistance? There's steps that we can take, hopefully, to kind of slow resistance down. But I think we're a lot more cognizant about this now, but in the past, maybe doctors have prescribed antibiotics when they knew it really wasn't going to do any good, maybe for an infection that was viral. Patients demand some treatment, so doctors are under pressure to do that. Sometimes patients don't take the whole dose. So you take maybe a few days and then you stop, and we think that can contribute to bacteria developing resistance. They've been massively overused in agriculture. and that's a hard problem because it really needs some kind of regulation to say that nobody can do it anymore. Because when you put antibiotics in animal feed, the animals typically will grow faster. And, you know, there's not conclusive proof that this is contributing to problems with human pathogens, but it's likely that it is. And worldwide, in other countries, some other countries, you don't need a prescription to get an antibiotic. So patients, you know, buy them in the pharmacy and maybe take the wrong dose, or again, don't take them as long as they're supposed to. So we think that that can contribute. Okay, so what can we do? You yourself always take your full prescription and encourage people that you know to do that. I know that my dad is really bad about this. He gets gum infections a lot, and he will, you know, take most of the prescription and then save a couple doses for the next time he gets a gum infection, which is, you really got to make sure that you kill all those bacteria if you can. Of course, if you're having some kind of side effect that you think might be due to the drug, then you should stop it and call your doctor and maybe get on a different kind of antibiotic. That would be the one exception to that. Whenever possible, we want to not take antibiotics. And that's kind of a tough one. Sometimes I see on Facebook, like people will say, oh, I think I have strep throat, I'm just going to gargle with vinegar. No, I wouldn't encourage you to do that. If a person does have a strep infection, there can be really bad consequences to having a kind of partially treated or untreated strep infection, things like scarlet fever or rheumatic fever. So I'm not saying that, you know, if you're sick, you know, with a bacterial infection, don't take an antibiotic. But there are some infections, like ear infections, that almost always clear up on their own. That's a tough one because that can be really painful. But, you know, whenever possible, we want to try to limit our use. Sometimes when we treat with two different drugs, you know, even if a bacteria sort of becomes resistant to one drug, you hit it with another drug and sometimes you can knock it out. We need to keep doing research and developing new antibiotics. You know, if we just can alter the side chain of it, then, you know, we have the drug, we know it's safe, and that can take not as much research to do that, but also looking into ways to target very specific targets. A speaker came to Grand Valley a few years ago that was experimenting with Mycobacterium tuberculosis and finding, you know, proteins that only that organism had and trying to develop drugs that would target that protein. So, you know, we can't avoid it. We try to slow it down. If we look at kind of zero in now on what the organisms are actually doing to make them able to survive in the presence of antibiotics. So there's several specific mechanisms, and this can be a little confusing, so don't be afraid. There's some more examples in OpenStack's textbook, so it's not a very long section, so I'd encourage you to read that part. But the first mechanism is that bacteria acquire the ability to just prevent exposure to the drug. Okay, so either the drug can't get in to begin with, or they are able to basically pump the drug out when it does enter the cell. Okay, so sometimes this happens just by point mutation, or they can alter the porin structure. So remember what porins are. They're little channels in that outer membrane that gram-negatives have for transport through that outer membrane. Sometimes just a little point mutation or two can make them specific and not let certain things in. Okay, so they're essentially decreasing that outer membrane permeability. And you know, the less drug that gets inside, the better chances that the cell is going to survive. Some organisms already have these efflux pumps. If they're exposed to some toxin, they have these pumps to kind of, again, limit their exposure. They can be altered to recognize an antibiotic, okay? If you remember, if you're in lab, when we tested Pseudomonas for resistance to antibiotics, that organism was growing right up to all those antibiotic discs. Okay, Pseudomonas just naturally has these efflux pumps to be able to, again, expel toxins. And we think that's the way that some organisms become multi-drug resistant. Okay, so that takes energy to do that, but it's not great. Mechanism 2, this is one that we've talked about before. Bacteria acquire the ability to make an enzyme like beta-lactamase. So remember what beta-lactamase does, it cleaves that beta-lactam ring right here, opens that up. So now it no longer is going to fit in the active site of that transpeptidase enzyme. Okay, so it's inactivating the drug. The bacteria is making the enzyme to inactivate the drug. There's other examples of this. I had to kind of scan the literature to look for that a little bit. But here's an example. Here's a drug, an aminoglycoside called gentamicin. And it's kind of, this picture isn't great here. It doesn't come through very well. But some bacteria acquire the ability to make an enzyme that might just change one of the functional groups on that drug. Like it's, one example was it phosphorylates some of these hydroxyl groups, or it can change and add, methylate a group, something like that. Just a little change to the drug so that it can't bind to the small subunit of the ribosome where it normally, okay? So causes inactivation. In other words, it can't bind to its target structure. Okay. Third mechanism, modification or protection of the target cell structure. So the bacteria is changing its own structure just slightly, not enough to impair its function, but enough so that the drug can't bind to it. Okay, so a couple of examples, ribosomal RNA methylation. So remember, that's a methyl group, CH3 group. It might just methylates a little bit to the ribosomal RNA. And again, so now the drug can't bind. Okay, so it's not methylating the drug, it's methylating itself. Okay. Another example of this is just maybe a very, maybe just a point mutation in the gene that encodes the target enzyme. So sometimes that could make the enzyme maybe not functional, but it could be a part of the enzyme that's not kind of going to make it non-functional. It's just going to make it so the drug can't bind to it. So for instance, maybe a point mutation in DNA gyrase would make it so the quinolones or the fluoroquinolones couldn't bind to that enzyme anymore. Or a point mutation in RNA polymerase, so the protein can't bind to it. And a couple more, sometimes, and I don't have a really good example of this. I think your book gives a little bit more examples, but target overproduction or enzymatic bypass. So if you've got an antibiotic, say, that targets RNA polymerase, for example, maybe the cell just needs to make many copies of RNA polymerase to make it so, the drug can't bind to all of them. Okay, or maybe there's some way that the organism doesn't actually need the enzyme anymore. It might find a way around that. Okay, and the last one, this is fairly newly described, but sometimes the bacteria can make proteins that actually prevent the drug from reaching the target. So mimicry, it's mimicking the target in a way to kind of basically distract the antibiotic from binding to the true target. Okay, so that gets a little bit complicated. Let's take a couple minutes and see if we can kind of think about that. I like this little exercise. So here's 5 different mechanisms. See if you can put, and I don't have them in order here. So see if you can decide, say, oh, number 3, which one of those 5 is this describing? Okay, so take a minute and maybe that will help understand how I'm describing these. So I'll give you the answers in just a couple minutes. Okay, I don't take too much time here, so let's go over this. I know sometimes it's just kind of, again, confusing who's doing what here, and even this drawing is a little bit confusing, but so this first one, efflux pump modification, that's if the circle here is the antibiotic and that's getting pumped outside of the cell. So that's number 3. Bacteria decrease their membrane permeability. So in other words, the antibiotic can't get in the bacteria. So that's number 1. If we modify the binding sites of bacterial enzymes. Okay, so here it's showing the drug binding to some enzyme in the active site. And here that enzyme is modified so that the antibiotic can't bind anymore. Okay, so that's number 5. Bacteria alter their porin structure. Here, these are the porins where the antibiotic used to be able to come in, and now it's not. That's number 2. And then bacteria modify the drug itself. So that's what this is trying to show, that the drug here now is altered. So that's number 4. Okay. Any questions about antibiotics? There's a really interesting, has anybody read the novel Aerosmith? It's one of those kind of classics, but that was written like in the early 1900s, and the whole, it follows the career of a doctor and how he started doing research in ways to use viruses to kill bacteria infections. And he was trying to cure bubonic plague by viruses. So he was way ahead of his time. It's an interesting novel, you know, if you're interested in this kind of stuff. But, you know, that's been proposed as, you know, maybe we're going to have to start relying on bacteriophage to treat bacterial infections. And there's work going on trying to do that. Okay, so yes. Okay, so thinking about next exam, which remember is a week from Friday, I am hoping to get through chapter 14 here today and Friday, but whatever I get through on Friday, I'm going to try to cut that off. And then next Wednesday's lecture won't be included on Friday's exam. So I'm hoping to get through 14. Right now I have 15 as part of the homework. So I'll probably push that off because I don't think I'm going to get to that. But okay. All right, so I'll put that in writing here at some point as we get closer. All right, so chapter 14 really and 15 both are describing the process of infection and what microbes need to do to be successful to establish an infection. So I I use a lot of examples of diseases. Okay, don't worry about the individual diseases for now, just concentrate on the principle that I'm talking about. So we will get to some specific infections later on where you'll need to know more about them. But for now, if I talk about the bubonic plague, don't worry about that. Just, again, the principle that I'm trying to illustrate. And lots of definitions here. The good news is hopefully a lot of the things that I'm defining at least look partially familiar. So we will do a lot of definitions. But first, I want to talk a little bit about our microbiome and how that we know plays a role in our overall health. We'll talk more coming up in the next few weeks when we talk about host immunity, how we think our normal microbiota plays a role in fighting off agents that might be causing disease. But what kind of organisms are part of our normal microbiota? I've been kind of used to just saying bacteria, but it's actually not just bacteria that we have living in us. We also have viruses. We also have archaea. We also have protozoa, as well as fungi. Okay, so lots of different kinds of organisms can be part of our normal microbiota. And of course, people are slightly different, or sometimes really different as to what their makeup of their microbiome. But it's been estimated that you have probably about 10 times more microorganisms in your body than you do have your cells of your own. So you're only 10% you in terms of number of cells. Of course, those cells are a lot smaller than our cells, but we can describe our relationship to these microorganisms as either mutualistic or commensalistic. So if we say they're part of our normal microbiome, then we kind of assume that they're not harming our health. They're just kind of there, hopefully helping us. If it's a mutualistic relationship, that means both organisms benefit from the association. And there's lots of examples of that. We know that the E. coli in our large intestine, we give the E. coli lots of nutrients, and in turn, the E. coli helps us digest food and produces some vitamins for us, maybe some folic acid. A commensalistic relationship is when one organism benefits and the other is not affected. And that's a little bit harder to come up with examples there. We used to say the staph epidermidis that is growing on our skin, we would call that a commensalistic relationship because we're giving the staph epidermidis all these nutrients. Well, what's the staph doing for us? Probably nothing, but now we kind of understand that the staph is probably out competing other bacteria that might be harmful to us. So actually that might be more of a mutualistic relationship. So sometimes we kind of learn more and then have to adjust our terminology. A lot of our microbiome could be considered transients, meaning it's associated with us for a time. We'll say a short time. You could potentially have whooping cough, that bacteria associated with your upper respiratory tract. Not enough to make you sick, but it could be enough that when you're around a baby to actually give the baby whooping cough. So something like that could be just transiently associated. Some of our microbiome, though, are considered residents. You acquire them soon after birth, and then you have those organisms for life. And of course, there's a lot of questions about, well, when we use antibiotics, how does that affect our microbiome? There's worry that especially when young children are given course after course of antibiotics, that disrupts their resident microbiome. How soon does it come back? Does it ever? Okay. Just as sort of, if you're interested, this is a table from our old textbook. If you're interested in the kinds of organisms that might be in your body, this is just a little, snippet of the sorts of organisms you might see there. Might see some fungi in your upper respiratory tract, a lot of different bacteria. Again, there's variation between people for sure, but there's certainly sites in our body that are considered azenic, meaning there shouldn't be any normal microbiota there. So places like our internal organisms or organs like our pancreas or our hearts, our bloodstream, as far as we know, should be azenic. That if there are organisms there, then that's a problem. They're causing disease. Okay. So let's start with some definitions. Question. Yes, so we'll mention that, but if somebody's doing chemotherapy, it's knocking down some of their innate immune cells, and they're more susceptible to a lot of different infections. Usually not their normal microbiota, but things from outside might be more likely to kind of have a foothold. Okay, so make sure we're kind of square away on some definitions here. Okay. All right, if some kind of organism, infectious agent is causing injury to the host, we would call that disease. And I should say that doesn't have to be an infectious agent. It could be a genetic origin, it could be an autoimmune disease, anything that's causing, again, injury to the host, we would call that disease. And when a pathogen is establishing and multiplying itself within a host, then we call that an infection. Okay, so usually when you have an infection, it's going to cause disease, not necessarily. There are some infections that maybe wouldn't. Typically, our microbiota, we don't usually refer to that as an infection, though, because that's just kind of always there. Of course, a disease-causing microbe, we'll call that a pathogen. And usually that's either a virus or a bacteria. Usually, if that disease-causing microbe is a fungi or a protozoa, we refer to it as a parasite. But I saw that the book is not really consistent there. It called Plasmodium, the protozoa that causes malaria, a pathogen. So, objective and measurable consequence of infection. We would call that a sign. Okay. For example, maybe fever, a rash, diarrhea, vomiting. Okay, we can measure that and see that when a patient is exhibiting those signs. Results of infection or disease felt by the patient, we call that a symptom. Okay, so we don't always use those two terms correctly, signs and symptoms, but an example of a symptom would be something like malaise, tiredness, or some kind of pain, headache. Okay, the patient reports having them, but as a physician, you wouldn't be able to measure that very easily. And then some terms when we're talking about disease. Number of cases of a disease, we refer to that as morbidity. And then the number of deaths due to disease is mortality. Okay, we're gonna explore the morbidity and mortality weekly report that's put out by the CDC every week, where it's a document describing, what diseases are being reported by doctors all around the country. An infectious disease that's capable of spreading to other people, we would call that communicable. And that's sometimes used in the same way that the next term, if it's easily spread from person to person, we would call it contagious. Okay, so again, you're kind of splitting hairs a little bit. Some infectious diseases are not communicable. Something like anthrax, you'd have to try really hard to infect somebody else with cutaneous anthrax. You would have to like rub your spores from your sore into somebody else's. So most of the time infectious diseases are communicable, but not always. And if they're easily spread, something like COVID, then it's going to be contagious. Diseases contracted as a result of a medical procedure, iatrogenic. I just had a friend who had a knee replacement done about 5 years ago. She just had to go into the hospital for an infection in her knee. So even though it was so long ago, just now it started causing problems. So that was a iatrogenic infection as a result of a knee replacement. Diseases acquired in a hospital setting, nosocomial. You've heard that term before. And diseases passed from animals to human. Those are zoonotic infections or zoonoses. So a lot of terms. We'll keep going there next time. We're out of time. So have a good day.